Our third class of biomolecules is nucleic acids, including nucleotides as the monomeric form and the polymeric forms of RNA and DNA. As we saw multiple levels of structure in protein, we see the same in nucleic acids. The primary structure in nucleic acids is the order of bases in the sequence. We learned in an early lesson that the sequence is informational, and in the case of nucleic acid, this primary structure specifies the genetic code. In this class of biomolecules, the secondary structure is related to hydrogen bonding, as in protein structure, and determines the three-dimensional conformation of the backbone. Tertiary structure relates to supercoiling, and the quaternary structure relates to interactions with other macromolecules which assist in transferring and interpreting the genetic code. Each nucleotide carries a characteristic nitrogenous base that is aromatic in chemical makeup. It is the identity of the base that distinguishes the different nucleotides. We can classify these bases into two categories depending on their structures. Pyrimidine bases are six-membered aromatic rings and are so called because of their resemblance to the chemical structure of pyrimidine. As pictured below, these include the bases cytosine in both DNA and RNA, thymine in DNA and in some RNA, and uracil found only in RNA molecules. Purine bases also contain a six-membered aromatic ring, but it is fused to a five-membered ring and is therefore a more complex structure. These include adenine and guanine, and each of these may be found in both DNA and RNA. As you can see from the structures below, they resemble the nitrogenous base purine. When one of these bases is joined to a form of the sugar ribose, it becomes a nucleoside, as depicted below. Notice the numbering system for the atoms in the nucleoside. The atoms in the base portion are integers, and those in the sugar include a prime symbol. This is how we distinguish the atoms in the sugar from those in the base. A ribonucleoside contains the sugar ribose on the left, whereas a deoxyribonucleoside contains deoxyribose, lacking an oxygen at the 2' position, shown on the right. Nucleotides, the building blocks of nucleic acid, are nucleosides that also contain one or more phosphate groups, as shown below. Note that the phosphate is esterified to the 5' hydroxyl group of the sugar moiety, and that nucleotides are named for the parent nucleoside. On the left we have adenosine 5' monophosphate because the sugar is ribose. On the right we have deoxyadenosine 5' monophosphate because the sugar is deoxyribose. Nucleotides containing two phosphate groups at the 5' position are referred to as diphosphates, and those with three phosphates are referred to as triphosphates. These monomers are the building blocks used in DNA and RNA polymers, which we will look at more particularly in our next lesson.